Bom dia, boa tarde, boa noite aos internautas que nos acompanham neste programa Nanoalerta, hoje no seu número 214. Fazemos este programa dedicado ao público não especialista, porque é este público que paga as pesquisas sobre nanotecnologia realizadas no Brasil. Também fazemos este programa porque é aqui que nós podemos discutir os impactos da nanotecnologia na sociedade, no meio ambiente, na economia, na ética, no direito, na saúde, coisa que não são discutidas aonde se faz nanotecnologia no Brasil. Por fim, fazemos este programa porque acreditamos que ser livre é ser bem informado e a divulgação científica contribui, colabora para o aprofundamento da cidadania e da democracia no Brasil. Hoje, conforme tínhamos anunciado, estamos conectados com o Dr. Arnan, diretamente da cidade de Melbourne, na Austrália, e que ele vem trabalhando com as questões de micro e nanotecnologia. Então vai ser este o nosso interação com ele hoje. Eu vou pedir para ele se apresentar e depois iniciar a apresentação dele. Ok, Arnan, could you please introduce yourself?
Thank you, Carlo, for inviting me to be on this program. I'm Professor Arnan from RMIT University in Melbourne. I'm a researcher, I'm director of RMIT's Micro-Nano Research Facility, a facility that I'll introduce in my slides, focusing on new nanotechnologies. And my own research area is integrated circuits using light instead of electricity, and I'll introduce those in my slides as well, and I'll talk a little bit about how we're using these for applications as diverse as testing pollutants in seawater, and also for detecting whether people have had a heart attack to protect them from the dangers of heart attack.
Okay, you can start your presentation.
Yep. Let's see if I can do this.
I think it's share.
Okay. Can you see my presentation?
Yes, yes.
Okay. So, in outline, I'll talk a little bit about the facility that I direct, and then I'll talk about optofluidic biosensors, so sensors that we're able to make using the nanotechnologies that we work on. And then I'll talk a little bit. I understand that this audience is not very technical, so I'll try and keep it as accessible as possible. So I'll introduce sensors a little bit, and then talk about how we use some of our integrated circuit technologies to make these new sensors. And then I'll talk about two specific examples that we've done. So, the facility that I direct at RMIT University, RMIT University has been working in micro and nanotechnologies since about 1983, so we have a lot of experience, but the equipment that was originally installed in 1983 was getting quite old, and so RMIT invested about $50 million Australian in this new facility, and it's the whole floor of a building. It has some very sophisticated laboratories that are clean rooms, and you can see perhaps here that there are people that you have to wear a spacesuit in order to be in this facility, to keep it clean, to prevent hair and any other dust from getting into the facility. We have maybe 250 significant tools in that facility, and the area where you have to wear this spacesuit is about 2,000 square meters, so as big as a large house. The major areas that we do research in are materials engineering, so creating new materials, some particularly new two-dimensional materials for sensing, biomedical devices, and photonic chips. And I'll talk in this talk specifically about the work we do on making biomedical devices that use photonic chips.
Very good.
I'll just skip over this slide. This is just the floor print of the facility. But the sorts of things that we make, essentially what the facility does is it's mostly for printing things. So printing structures in sort of 2 dimensions, but at very, very small scales. So some of the biggest things we do are up here, and this is actually a chip that we designed that is a artificial mosquito stomach. So this is a there's an environment here that has all these little pillars in it, and it's actually designed for these little parasites, so to emulate malaria parasites, to live in this environment. But this, for us, these are about 5 micrometers, and just for reference, your hair is about 100 micrometers, so this is about 20 times smaller than the width of a hair. But for us, that's very, very big. So if we move down to the next size of objects, we tend to make structures mostly that are about 1 micron. So these structures here, you can see that's actually half a micron. So this distance from here to here is 1 micron, and that's 100 times smaller than a hair. But we can actually print these circles here that you can see at that very, very small scale. Then if you want to get really small, we can sort of print these sorts of structures, and for example, these triangles that you can see here, the gaps in between those triangles are about maybe 50 nanometers. Now, an atom is about a little less than 1 nanometer, or a molecule. So the very basic building blocks of matter, this is really only about 50 of those across. So this is very, very small indeed. But the smallest tool we have for patterning in our facility can actually achieve resolutions of 1 nanometer, but they're too small to see, so you can actually manipulate matter. This is actually a 3D printer that we have, and the features of this 3D printer are about 100 nanometers, and this is actually too small to see with light. You can't see it even under a microscope. So we have to use an electron microscope to be able to see this. So these are just some of the examples of the sorts of things that we make in the facility. And as I mentioned here, so these structures are about as big as we get. So we start getting uncomfortable about the size of a hair, about 100 microns. So what we've done is we've actually invested in another facility, which you can see here. This is a low temperature co-fired ceramic facility, and this is really designed for making structures that you would put microchips in. So some of the things that we've patented that are very small dimensions, we would put those inside these things that are about, you know, big enough to pick up, but still quite fine featured. And they're made out of ceramic, or like the same sort of material that you make pottery out of. But we again print and pattern those with sort of circuitry, as you can see here. So these are some of the tools that we have used, and this is generously supported by the Australian Government research infrastructure funds. And the sorts of things that you can make with these sorts of technologies, these aren't things that we've made ourselves. These are a commercial company, Kyocera has made them. But there are particularly electronic circuits, like very fine electronic circuits. You can see here, this is sort of the packaging that you would normally find a computer chip to be packaged in. So this is, you know, if you pick up a computer chip, what you're actually looking at is a ceramic package. So we can make those at RMIT. But you can see here, these are the sorts of packages that we would be looking at for packaging chips in these sorts of boxes that are very rugged and environmentally stable. But we can also make fluidic channels. And actually this example here is some work that has been done with our collaborator, Mario Rubio, who works at IPT in Sao Paulo. So there's a connection there to Brazil. So my own research is actually in photonic chips. So this is an example of a photonic chip. And this is made out of silicon, the same material that electronic chips and computer chips are made out of, but instead of having electricity on them, the chips I work on have light running through them. So there's you couple light in through this structure, and it travels along the waveguide here. And we do a lot of work on designing these chips, and actually we have made some software that is used globally to design these chips and help fabricate them. But we also work on interfacing these chips with fluids. And so, for example, there's an example here where we've worked with the University of Ghent in Belgium to interface these chips to liquid crystals so we can control the light going through the chips. But we've also looked at connecting these chips to some microfluidics for doing testing. And this particular chip, this chip actually was some work done with our collaborator, Professor Laura Lechuga, who's based in Barcelona, but she also is a lecturer, I think, with a professor working with Unicamp in Sao Paulo. This what we did was we worked with her to get this chip and made this automatic microfluidic, so a sort of integrated circuit that deals with water. And this chip actually samples seawater. It sits on a buoy floating in the Mediterranean Sea off the coast of Barcelona, and measures seawater and tells whether there's antibiotic contaminants that might have come from farming down the river into the ocean. So we also actually provide a service with our facility for fabricating photonic chips. So you can see here there is a design of a photonic chip, and here there's the actual chip that was made. So if we've provided a design, we can very quickly make a chip, and this is work that we do in collaboration with the IMEC in Belgium and also the University of Ghent. And we also, as I mentioned before, have done a lot of work with the University of Ghent to make tools that you can use to design these sorts of chips and have them made in factories around the world. So now I'm going to talk a bit more generally about biosensors. And so what we're interested in trying to do is create tools that you can use to diagnose human conditions. So for example, pregnancy, you can measure hormones to various different hormones that mark pregnancy. You can measure blood poisoning, sepsis, by detecting bacterial markers. You can detect whether people are having a heart attack or whether they've had a heart attack recently. And this can be very important. If someone comes to a hospital and they have chest pain, it's important to know if they're having a heart attack or if it's indigestion, because if it's a heart attack, they need to have surgery very quickly. But many people present with chest pain, but only 8% of them are really having a heart attack. And there's also a lot of interest in trying to detect markers of cancer. And so we can screen for a very early detection of cancer and prevent this disease. So what we need for this is very, very selective detection. So we want to be able to detect some very specific biomarkers here to detect these different human conditions. They need to be very, very sensitive. So, for example, we want to take a drop of blood and detect very, very few of these biomarkers. So, for example, the cancer cells, there may be only 1 or 2 of these in a whole sample, and so we need to be able to detect very, very small quantities. And we need to be conclusive. We really need to be able to not just measure are they there or not, but actually measure exactly how much is there so that we can make a formal diagnosis and actually act on it. So for example, with the heart attack, we need to know whether that person's having a heart attack, when it started, and in order to know when it started, we need to be quantitative, and that will actually determine whether or not that person should have surgery. And so it's a really life and death decision, so it's very important. So the types of biomarker sensors, a pregnancy biomarker, these are very common as a lateral flow device. You can buy them in a chemist, but they're selective, but they're not really conclusive. If you get a positive on this, then you still have to go and see a doctor. You're not going to make clinical decisions based on this. They don't have to be very sensitive because the hormone that they're detecting is actually, there's a lot of it in a urine sample. It's not urgent. So you can wait a certain amount of time to get the answer. It's not quantitative, it's sort of yes or no, and the cost is pretty cheap. For a heart attack, it needs to be very selective, and you want to be conclusive so you can determine treatment. It needs to be very, very sensitive. There's really a very small number of markers and they're very small molecules in blood. It is very urgent, so you need to take blood, and blood is very complicated. Urine is quite simple, but blood is very, very complicated. There's a lot of different things in blood, the diagnosis and treatment depends on the quantitative levels, and because of the significance and the urgency, the cost of this can be higher. And so this device is essentially a piece of paper that's been sort of treated with a few antibodies. This usually you have to have full blood work and go to a clinic, but that can be too slow, and so it can actually take time, which might cost a life. The blood test requires plasma extraction, so you can see here a centrifuge extracting plasma. It's a slow time to get sample to the clinic to process with the centrifuge. It requires infrastructure and expertise, so you need to know how to run one of these machines, and you've got to have one of these machines on site. And there's a move to having patients initially treated rather than going straight to the emergency ward in a hospital, to have them treated in a sort of a clinic, and that may not have a centrifuge and this sort of blood work infrastructure. And the biomarkers can change over time during processing, so you don't really want to process them too much. So what we'd like to do, and what we're working on, is trying to create a solution that you can actually send out in an ambulance, so it can detect the type and severity of a heart attack, and it can be in the ambulance, or it can be in a satellite clinic, one of these, not a hospital, but a clinic that's sort of intermediate.
Yes.
So it's got to be compact, self-contained, relatively low cost, particularly have low cost consumables, be automated. You don't want to have a lot of skill to operate this. You want to be able to have it operated by a paramedic, for example, in an ambulance. And it needs to be reliable, robust, and if there's going to be a lot of them, it needs to be manufacturable. For clinical diagnosis, it needs to be very sensitive to detect very low concentrations, and very selective, so that it can really determine exactly what it's measuring conclusively, so that you can make life and death decisions about surgery based on it. So the sensors that we work with are generally immunoassays, and what that means is that the sensitivity is actually achieved using antibodies, and often the antibodies will come from an animal, and these are developed to respond to particular biomarkers. So, for example, for a heart attack, there's a protein that our heart muscles use to contract called troponin, and you can actually train antibodies in, for example, a rabbit or a goat to respond to those, and then use those antibodies to actually be your sensor. So this is a picture of an antibody. This is sort of the symbolic representation of an antibody, and this is, you know, generally the symbol of an antibody. And essentially what it does is it has all these very complicated sites on it, but only very specific antigens, things that it will respond to, will actually be able to stick onto those areas. So you can see here these different symbolic shapes, but only one of those shapes actually matches the shape that's on the antibody. So this is sort of chemically what an antibody does. And so if you can coat your sensor with these antibodies, then you've now got a selective tool for detecting these things. And you can purchase antibodies for lots of different types of biomarkers. There's a lot of biochemists who've created these antibodies. So in a clinic, generally what you have is what's called a ELISA, and this is basically you have the thing you're trying to detect is immobilized on the surface. You have your antibodies with some chemical attached to them. They stick to the thing you're trying to detect, and then that chemical that's attached to the antibody makes these objects fluoresce, so you can sort of see where this antibody is by it lighting up. This is a really good test, and it's very quantitative and selective, but it requires really, really careful sample preparation, so it's got to be done by a specialist. You need lots of reagents, and you need to use a microscope to actually detect this. So this isn't really suitable for an ambulance. So these are the sorts of tools that are used in the clinic for actually doing these tests, but it's not really suitable for an ambulance. So what we'd really like to do is take this tool and turn it into a chip that's about the size of a 1 cent piece here. So this is now some work that has been done in collaboration with a colleague, Laura Lechuga, and this is the work that she published here. But what we can see is that if you imagine you've got light that is trapped in this film here, and there's total internal reflection, so the light is bouncing back and forth between these two walls, some of the light will actually see these antibodies, and so as the things that we're trying to detect start getting stuck to the antibodies here, the light will actually see these a little bit and will slow down. And so if we can detect the light slowing down, then we've got a very sensitive way of determining the antibodies and how they're responding on the surface. And so the way that you make these structures is that essentially you have like these wires for light. They're called optical waveguides, and you have this structure here, you put the light in, it gets split in half, so half the light goes through here and half the light goes through here, and then you recombine it, and if the light was delayed by the same amount in each arm, then you get interference between the light, and if the light is in phase in both arms, then it will add up positively and all the light will come out here. And if this light is slowed down enough to then it will interfere destructively with the light here, and it'll be dark here. So depending on how much of the thing you're trying to detect accumulates on the surface here is whether you get a bright or dark response here. And this is what's illustrated here, is that you can see that as the analyte is accumulating, the light increases and decreases. So this is actually a chip that we made at RMIT, and you can see this is actually the region where the light is exposed to the blood, and so the light comes along here, goes around the spiral and interacts with the blood, and then comes back here. And meanwhile, the other half of the light just goes through this path, and then it recombines here. And if there was a lot of the material that we're looking for in the blood, then this will be slowed down and it will switch the light off coming out here. And that, again, this is what we see here, is the light going on and off depending on material arriving at the surface here. So this is a very, very sensitive way of detecting chemicals, and its potential, if you design this right, you can actually get to the point where you're detecting even just a single molecule of the material that you're looking for is possible to detect using this sort of technique. However, you need a really, really good quality laser and a very, very good quality sort of tools, like laboratory tools, to measure it, and that might not be possible to have in the back of an ambulance. So there's a different approach called surface plasmon resonance, and here you basically shine a light source onto a metal film, and the light can sort of get trapped in that metal film if it's at a very particular angle of incidence. And so you can see here a laser beam illuminating this metal film, and one of the angles here is dark because that angle got trapped in the metal film. And the angle that gets trapped here, and so what you can see is on a readout, if you look at the angles on this detector, you'll see a lot of these angles are bright, but then one of them's dark. Now, if there's material on the other side of the metal, that actually changes the angle of light that gets trapped in the metal. So you can see here, if these antibodies attach to the surface, then the dark spot here moves across, and if you can measure how much that dark spot moves, then you know how much material has arrived at the surface here. And so that's how these sorts of sensors work. And so here's an actual measurement from one of these sensors, and what you can see is that if you look at the angle that the light came in at, then you can see that at some angles it's bright, for this angle it's dark, and then for this angle it sort of gets bright again. But the dark angle, as you actually have different materials attaching to the surface, the angle actually changes, and if you can measure that change in angle, you know which materials have attached to the surface. So this is the basis actually of a very common instrument in bioanalysis called a biocore. This is made by General Electric. But the size of this tool is it's about 1 meter wide, and it costs about $100,000, and it's got lots of moving parts and really probably also requires a fair amount of skill to operate. So this really isn't a point of care device. So here's an alternative that we've been working on with another of our collaborators. This is based on basically a metal film, and it's got lots of holes punched out. And by punching all these holes in it with different spacing between them, and these holes are very, very small. These holes, you can see this is 1 micron, so that's 100 times smaller than a hair. The holes are about 200 nanometers, so about 1/5 of that. So again, too small really to see with light, but the holes and the spacing between the holes can actually significantly change the color of light that is transmitted through the holes. And I'll show you a picture of this. So they're too small to transmit light directly, but for some wavelengths you'll actually see the light that gets affected by the holes can actually pass through. And so here's a really nice example of some structures that you can see, and these come from this paper here, that all of these colours that you see aren't really the colour of the material. They're formed by having these very tiny holes in the metal film, and by controlling the size of the holes and the spacing between the holes, you actually change the colour that gets transmitted through them. And so here's another example. This is actually an optical image. You can see a red and a green, but actually it was the same colour light that went through them. It's just, if you look here at an electron microscope image, you can see all the little holes here. And the difference in the size of the holes between this one and this one actually makes these red and these green. So this is a really nice way of making a very simple optical sensor, because you can just have a white light source. So you can just have a very simple light emitting diode, a white light source, and just like a camera, like your iPhone camera, and then put these metal sheets with all these holes in them here, and then just put them in between and just measure the colour going through. And then you can have fluid running over the top, and if material sticks to the surface here, then the colour that gets passed through here will change. So this is some work that we're doing in collaboration with some researchers at EPFL in Switzerland, and what they've got is these metal sheets with holes drilled in them, and these antibodies attached to the surface, and if the target attaches to the antibodies, it'll actually change the colour that goes through this sensor. So if you look at this response, for example, you can see time in minutes, so it's about 30 minutes. As you add the analyte, the colour here, the wavelength, actually changes from this colour to a slightly different colour, and you can measure that change in colour. So we have some of the bits of an integrated point of care chip. We have this structure, this sort of reader tool. We can either measure using waveguides, using Mach-Zehnder optical waveguides, a photonic chip, or we can measure using these sort of little holes, nano holes in a metal film. But what we'd also like to do is integrate these components, the assay and the sample preparation. So for that we use, rather than photonics, we use microfluidics. So here's some pictures of some chips that have actually been made for controlling fluids on an integrated circuit. And you can have, these are often made out of plastic or glass, or also silicone rubber. This is polydimethylsiloxane, but essentially this is the same as silicone rubber, like silicone sealant that you would find in a bathroom. And you can make very, very complex systems with thousands of valves. So this is a really good example of something with thousands of components on it. So this is my PhD student, Crispin Zidzig, and he's been working on a very simple technique for making microfluidic chips, which is essentially an injection molding technique. So he has two plastic structures with features on them. He presses those together, and then here he injects this liquid silicone rubber in between, then heats it up, and the silicone rubber solidifies and goes solid, and then you can sort of take the two mold parts apart and peel out this silicone rubber part from between. So this is a picture of one of the chips that we made, and the important thing is you can have channels on the top and channels on the bottom. And this is a structure where we've actually grown some cells in the middle, in this middle part. We can also make some valves, and maybe you can see this video running. So this is actually a tiny little pump that we have made on this chip. And so you can see we have all these different colors and some valves here that control which color goes in. And you can see that here, for example, we're pumping red in here and then red in here, and then we switch to yellow. So you can see the valve switching here to yellow, and then we're pumping yellow through here and yellow through here. Now, with this structure, we can actually have different reagents and our samples in these different chambers, and with a computer, we can control which fluids go to our sensor down here or the waste channel down here. So this chip was actually designed for the automated system that went on a buoy in the ocean near Barcelona. So this is an example of that chip, and we can make these very, very cheaply and easily. We can basically injection mold them and make hundreds of them quite cheaply. So this sort of changes how the complex study works. It can also help with some of the sample preparation. So this is the structure that we worked on with Professor Laura Lechuga. It's an optical waveguide. It has an opening in the top where there are antibodies attached to the surface, and as the light passes through here, the antibodies interact with the light and slow it down and give you either bright or dark transmission here. And if we can measure that, we can measure the amount of material that's attached to the surface.
I know this Professor Laura Lechuga.
Yes.
I was one seminar in Vigo, and Laura was there.
Yes, so Laura's a collaborator of ours, and she's actually one of the external advisory board members for our facility, and she visits Australia every year. She'll come and visit us in November this year. Yeah, so we have a very strong collaboration. One of her postdocs is in my lab at the moment working with us. So this is the system that they used to have, which is a bit complicated. And so my PhD student went and visited Laura for 6 months and replaced it with this system, which essentially has all of the components needed to do the assay all integrated into this chip that can be controlled. And so what you can see here is that we can automatically control all the different fluids going into this chip, measure the baseline, put the sample in, measure the sample getting collected by the antibodies on the surface, washing the sample out, cleaning the surface, and then basically resetting all of the antibodies by cleaning them off and getting back to the baseline here again. So you can sort of run the sensor over and over and over again. And this was important because if this sensor was going to sit on a buoy in the ocean, you need to be able to use it every day, but then clean it out and have it ready for the next day. So the second example, and I'm very close to the end now, is some work where we wanted to use these nanohull arrays. And so this is some work that we've done, that was done in collaboration with Hatice Altug at EPFL in Switzerland. And so essentially their system is just this metal plate with all these holes drilled in it that goes into this system here. It's illuminated by a white light source and picked up by a camera. And what you can see here is that if you have just the, you look at all the different colours, the different wavelengths of light, the different colours, you can see the black line here is what happens before the chemical you're looking for is applied, and then when the thing you're looking for is applied, you see that the colours that transmit change. So she was interested in doing this, but what she wanted to do was capture an individual cell here, have a very, very small channel, and look for very, very small quantities of chemicals that the cell was secreting, to talk to other cells. And the problem with this was that if you have a very small volume of fluid here, very, very quickly that fluid will heat up and will evaporate. And so it was very hard to keep the cell alive and keep the fluid around it stable over the time that we needed to do this measurement. So what we did is we came up with this system here, and this was to capture the cell. So the cell would flow in here and get captured here, and then we had these valves that we could lift up or drop here. So we have those open initially and then drop the valves here and here to trap the cell. And then we have this channel here that basically lets water at a sort of body temperature pass through here, and what that does is it keeps the liquid that the cell's in at the right temperature, so it keeps it at body temperature. But it also, this PDMS rubber is actually slightly porous to water vapor, so it's possible for fluid to evaporate through the PDMS rubber. So by having this constant fluid flow over the top here, we're able to make sure that the liquid, the humidity is kept constant, and the liquid here doesn't evaporate. So we were able to then operate this over even a day to make these very precise measurements on these cells. So here's actually a microscope image, and what you can see here is these are the valves that we use to capture the cell. Here's the cell that's been captured, and here's the channel that's sort of regulating the humidity and the temperature. If we zoom in, you can see here's the cell, and then zoom in, that's the cell and the channel there. And so we want to see what that one cell's doing. Now this was quite. This is actually a picture of that cell trapped on the nanohull array. This has just been published in the journal Small, which is all about micro and nanotechnologies. And we were lucky enough to get our work featured on the cover of this magazine. So this is about the ability to see individual cells, not only see the cells, but actually see the chemicals that they use to talk to each other. So, one of the things that's missing, we've talked about the assay, we've talked about the sophisticated breeder, but we also need to prepare the blood. So a lot of work that we've been doing right now is actually looking at circuits that we can put whole blood in that will actually separate blood into its components and be able to pump blood around. If you're not careful and you pump blood too hard, it will actually start to clot in the chip and change its nature. So what we want to do is be able to have the blood actually moving through the chip and be able to start separating it into its plasma and pulling the cells out of it so we can study them, but without actually changing the nature of the cells, and particularly not upsetting the platelets that cause blood to clot. So this is a lot of sort of my team has been doing, and in the journal Lab on a Chip. This is my final slide.
Yeah.
This is my final slide. So basically, talking about the facilities of MNRF, I've talked to you a little bit about integrated photonics and its use for biosensing. There's elements for optical interrogation, for automatic fluidic assays, and blood handling, which is the last thing I talked about. And I think really in Outlook, by integrating, we can certainly help a lot of our research colleagues, so they're very interested in being able to use our integrated circuits for doing biomedical research, but we actually think that this is really going to change the way that point of care applications are done, so whether people in a hospital or in an ambulance can actually use these devices. Thank you.
Okay, thank you so much for your contribution to our program. It's a very, very interesting research in many areas, no? That we can know more things now after your presentation. So I don't have enough knowledge to put some in-deep question for you. Okay, so I will put some more general questions.
Okay, that's okay. Yep.
And my first general question is about the time. How much time, how much year to do this kind of research?
So, honestly, from fundamental discovery right through to start, you can be talking about 12, but and do with accessible as possible. So, for example, you might remember me talking about that mosquito stomach we tried to make that was actually done by a final-year engineering student, and it was done in 6 months. So we had a collaborator who was interested in having this made, and they described the problem to us. So we came up with a solution and actually tested it in 6 months. So that's pretty fast, 6 months.
Okay, I don't have your image, but I have more question for you. About the cost to do this kind of research. What you can tell for us?
So it depends, and it depends what you're measuring in terms of the cost. The materials can be quite inexpensive. So let me see, can you... the PDMS material for the microfluidics, let me just get back to the picture that I showed you, right here. So, are you seeing my screen?
Yes.
Okay, so this material, the PDMS itself is about, it's less than $100 for 3 litres, so this is very, very inexpensive. And if you make a lot of these devices, you can injection mold them so they can be a few cents each. So this is quite inexpensive. And at the moment we've just installed a new tool which is like a laser printer for being able to print these sorts of chips. That tool cost us maybe $500,000. So that's expensive, but it doesn't really have any consumable parts, so we can use that over like 10 years and produce these chips in about 10 minutes using that tool. So these sorts of structures can be very, very inexpensive. These sorts of structures, it's much more expensive. Like if you wanted to have a very high resolution structure, if you want to make one of them, it can be, it costs $1,000. But and that's just in tool time. It's not even in the time it takes for a person to do the design. But if we go forward to the nanopolymerase that we were talking about with Hatice. So these sorts of chips that we work with Laura Lechuga, in principle you can make them for about $1 a chip, but you need to be making a lot of them. You really need to be making, 100,000 or 1 million of them in order to get them for $1 a chip. At the moment we charge about $1,000 to make one of those chips, to make them one at a time. And the same thing for these sorts of structures that we see here. These are very, very fine featured holes, and if you want to make them one at a time, it would be about $1,000. But if you can make millions of them at once, then they can be, maybe 10 cents each.
Okay, and your university is a public university or a private university?
It's a bit complicated. I mean, I think the short answer is that it's a public university, but actually it's non-profit. But it's actually private. So in Australia there aren't that many private universities. I know there's a big difference between public and private in Brazil.
Yeah.
In Australia, it's most of the universities are public, and RMIT I think by most measures is a public university, but I think technically it's private. It was sort of set up by a generous donation by an individual over 130 years ago.
And you have public funds to do this kind of research?
So the university has invested a lot in infrastructure and has been investing in hiring staff who are able to do research, but generally the funds we have to do these projects, we have to compete for external funding. So we have a lot of competitive funding that we've gotten from the Australian government through the Australian Research Council. But we also have a lot of projects that are funded through companies. There's a lot of biomedical research, particularly in Melbourne, and some of that's publicly funded. So our end users have government funding, but they pay us to sort of come up with the solutions for them.
Yes, you quote IPT here in São Paulo and the UNICAM.
Yep.
UNICAM, what is the research that is your partner?
So we've been building a collaboration, particularly in using sort of microstructured ceramics, and a researcher at IPT in Sao Paulo is Mario Rubio. He's actually regarded as a world expert on ceramic microstructures for microfluidics, and so he's come to visit our facility late last year, and another researcher, Adriano Alcalna from University of Sao Paulo has also come to visit us, and I've been to Sao Paulo several times to IPT to see the facility there. So the facility, the sort of micro and nano fabrication facility at IPT in Sao Paulo, it's actually very similar to the facility that we have at RMIT. There's a lot of parallels.
Yes, I know Mario Rubio, because I also worked in the IPT.
Okay, very good.
But it's, I am retired from 2012, so I see him. I saw him a very long time ago. But the last question that I would like to put for you is about the bottleneck. What is the bottleneck for this kind of research in your opinion?
Okay, Okay, so my personal opinion is that people thought that, like microelectronics, so microelectronics is a wildly successful micro and then nanotechnology, and people thought that photonics, and photonics is beginning to get there, would accelerate and become, very, very complex and sophisticated like electronics, and microfluidics would do it as well. And people have been predicting that for many years, but it hasn't really happened. Now, I believe that this is mostly due to the interfaces, and that basically if you need to have a lot of tubes, and I'll show you my picture again with all the tubes coming out of it.
Yeah.
So here you see structure here. There's a lot of tubes coming out of it, and the more complex this structure on chip gets, the more tubes you need to have coming out of it. And I think that's really the limitation. If we can get rid of all these tubes, or have one tube and have all the complexity in that one tube, that will be a major breakthrough and will allow things to become more complicated. So this is and the same thing with the photonic interfaces. You need to have lots of optical fibers, or the interfaces to the optical fiber, which is one of the beauties of this sort of setup, is that the interface here is very, very simple. You've really just got one LED light source and a camera, and then you just put the chip in, and there's no more interfacing to do. So I think really the bottleneck is mostly the interfaces, and trying to make those interfaces as parallel and as simple as possible is really the key.
Okay, thank you so much for your contribution in your time. That is very long nights, and I hope you will continue to do this kind of research, and when you have news about your research, you can do another program.
Okay, and yeah, feel free to ask me any questions. My contact details are available. I'd love to hear from anyone.
Okay, thank you so much. Have a good night.
Thank you. Okay, bye.
Bye.